Hello all, welcome back to Dr. Kala. Today we are going to discuss about recombination and genetic mapping. So before 2-3 videos back we were discussing about crossing over. So next to crossing over, what all things happens in this inside this chromosome and genes are said to be that recombination and genetic mapping. So regarding that we are going to discuss today. So crossing over, it is going to leads to combination of new characters. Okay, so crossing over, what it leads? It leads to combination of new characters, so which is called recombinance. So due to this combinations, we call it as recombinance. Okay. What happens in this crossing over is cutting and joining. Simply, if you are telling us cutting and joining, what happens? So like this. So what will happen? Here one cut and here one cut and then it get recombinant. Okay, so the alleles itself will get changed from one chromosome to the another sister chromosomes, sister chromatids. So hence this process is called recombination. Okay, so this combination of new characters is called recombination and total process is called as an recombination. So actually regarding recombination, a widely accepted model is Holiday Junction model. So what is this Holiday Junction model? So this is the name of a scientist, right? So he has proposed a model regarding this recombination, and this was widely accepted. What we spoke in crossing over and about the this calculation of recombinant frequency, everything is matched with this Holiday Junction. So in this Holiday Junction, what the scientists try to explain is, first, the DNA endonuclease. Endonuclease is the enzyme which is going to cut this particular part. Okay, it will cut and it helps to rejoin. So the junction which has got rejoining is called as an Holiday Junction. Okay, so we are not going to get into detail about that. So next we are coming about calculation of recombination frequency, which is called as RF. Okay, calculation of recombination frequency, which is called as RF. So we call it as percentage of recombinant progeny in a cross. So if an percentage, how many percentage of recombination has happened, has occurred in that particular progeny, in that particular cross, is called as an recombinance. So we can calculate it by number of recombinants divided by number of offsprings. So number of recombinants divided by number of offsprings. To any type of cross we can learn this. Any sort of cross we can learn about this recombinance. For example, what we can tell, in dihybrid cross we have a ratio of 9:3:3:1. So this is not a recombinant and this is not a recombinant. We all know that this is yellow round and this is another parent. Okay, so it is wrinkle green. Okay, wrinkle green and yellow round. So these are recombinants. So what we want to write? Number of recombinants is 3 divided by total number of offsprings. So we have 16 offsprings. So 13 divided by 6 into 100. This is going to give the percentage, which is the recombinant frequency of what? Dihybrid cross. Like that we can calculate it for so many crosses. What type of Mendelian crosses are there? All the crosses can be calculated by this simple formula. Okay, so next is genetic mapping. So what? Genetic mapping. This is the continuation of this. So Alfred H. Struvent, he is a student of Morgan. We have read about linkage, right? We have studied linkage. So regarding that, Morgan has proposed a lot of theories regarding that. So his student Alfred H. Struvent in 1913. So he only first put forward the concept. He only explained this concept regarding genetical mapping. So generally in social studies, what we will do? Mapping. So we will point out which place is there, Delhi, Chennai, Mumbai, like that. We will point out the places where it is. In the same way, in the gene mapping also, we are going to find the point where the particular gene is present and how much distance it is there between the genes. That is calculated in this gene mapping. So genes are presented in a linear order, that we all know commonly. In a linear order it is present. What we call the position where the gene is present is called as locus. Okay, this is singular. Locus means singular. Loci, if you tell loci, it is plural. Okay, so next is, what is genetic gene mapping? It is nothing but a diagrammatic representation of a particular position of a gene or a distance between the particular genes is calculated using this gene mapping. Okay, so this is also called as linkage map, because as we are calculating the distance, it is also called as linkage map. Okay, so actually this genetical map is directly proportional. This word is very, very important. It is directly proportional to the frequency of recombination. According to that frequency of recombinations only, these things are interdependent and it is directly proportional. And it provides clue. What for it is providing clue? It is giving the clues where the gene is hiding or where the gene is lying in that particular chromosome. We can tell that our skin color is wheat color. Can we tell where the genes is present in the for the gene color? That thing is identified using this gene mapping. So hence it provides clues where particular genes are located. And next is map distance. So in a genetical map, particular unit of distance is called as an map unit. In short form, it is m.u. Okay, unit of distance in a genetical map is map unit. And regarding this, one map unit is equivalent to 1% crossing over. What is that? One map unit is equivalent to 1% crossing over. So one map unit we can also call as centimorgan. Also, we can also call this one map unit as centimorgan. Okay. So if one map unit is called as 1 centimorgan, right? 1 centimorgan. And the next thing important is 100 centimorgan is equal to 1 morgan. 100 centimorgan is 1 morgan. So here centimorgan is represented by small c and m morgan. We all know a scientist's name. So it is capital M. So here morgan, capital M. You should remember all these symbols here. So okay, for example, if they are giving two genes, A and B genes, A and B genes, which is having a distance of 3.5. Okay, what 3.5? We can't tell centimeter, millimeter and all. Here we are calculating a centimorgan, 3.5 centimorgan. So how you will calculate for all these things? How you can compare with all these things? That is thing is, we can tell it as first thing, what is that? One map unit is also called as 1 centimorgan. So if here 3.5 centimorgan means we can represent it as 3.5 map unit, not this one, 3.5 map unit. So 1 map unit is equal to 1% crossing over. So 3.5 map unit is equal to 3.5% crossing over. Then according to this formula, this recombination formula, into 100. Okay. So crossing over is 1%. So recombination means we are going to get into 100. So if it is into 100, what will happen? 0.03 0.35% recombinance. Okay, the recombination frequency RF will be like this. So if they are giving anything, centimorgan or 1 map unit, this thing recombinant frequency or crossing over percentage, whatever they are giving, you should able to understand this and you should get answer to this. So 1 map unit is equal to 1% crossing over. 1 map unit is also called as 1 centimorgan, and 100 centimorgan is equal to 1 morgan. You are clear here. So next is uses of genetic mapping. So what are the uses of genetic mapping? Why we want to do genetic mapping? The first and foremost thing is to find the gene order, in which order it is present. What are the proteins expressing genes or character expressing genes, where it is present. For that determination, this gene mapping is very much useful. And second thing is to identify the locus of that particular gene. What is the locus? Locus means particular position where it is present in that linear order. For that it is very, very useful. The third thing is to calculate the distance between genes. As I said here, to find there are lots and lots of genes. We want to find where it is present, in how much distance it is present. For that this is very, very useful. Next thing is result prediction. As what I said here in recombination frequency, for dihybrid cross I have given the formula and we can find the recombination frequency. So like that, whatever genetical cross or Mendelian crosses are there, we can predict the results. For example, dihybrid, trihybrid, incomplete, what type of crosses also we can predict the results. And then overall genetic complexity. If an organism having an complexity, so that how the gene is getting expressed, how this genetical complexity will be there is getting explained by this genetical mapping. So I think you will be clear with this video. According to the examination point of view, here the map distance are very, very important. Regarding NEET also they used to ask this calculations, and it is very, very important. Uses of genetic mapping is a 3-mark question, and the thing is you need to know this recombination frequency formula. Everything I have explained here, so it's clear about recombination and genetical mapping. If you have any doubts, you can comment me in the comment section. I will clear your doubts. Meet you in the next video. Thank you.